All right, so still in the week of the 18th through the 21st, so we did intro A&P Tuesday, so we're going to cover the cell today. All right, so you should have exercise 3 homework to do. You should also have, I think, your post labs for 1 and 3 this week, I believe. Next week, we're going to do a kind of review session on Tuesday, and then you'll take the practical on Thursday. Okay, so hopefully we'll have time to actually go and walk through so you can do the practice practical, so that you can also do that on your own. All right, so let's go ahead and start the show. So we're going to start with cell anatomy. All right, so you have a few of these around the room. And you got 2 that are like this, 2 that are nerve cells. We got 1 over here, it's another cell. They're all cells, so they all have basically the same parts on the inside. So for the practical, you're gonna have your answer sheet and board. Hold your board up while you're walking around, write on it, write your answers. When you come to the station, it's gonna have a cell there and have a label and number on it. The question on the table will say, what is this structure? Or what does this structure do? And you write it down. So you need to be comfortable whether you're looking at that, or that, or that, so you know what the parts are. Okay? So we'll start with the nucleus. The nucleus, to, I guess, borrow from the, meme that's going around, it's where the DNA resides, right? So that's where your genetic material is. Now, inside the nucleus is the nucleolus. Okay? So nucleolus is where ribosomes are produced or created. Okay? A ribosomal RNA is made. So important piece of information you need to know in the nucleus. Again, is where the DNA is, and in the process of transcription, and I'll move out of the way so you can see it, DNA is used to make RNA in the nucleus, okay? Now, that RNA will leave the nucleus through nuclear pores, okay, little holes, and it goes through. Finds ribosomes, okay? Now, ribosomes can be free floating, or they can be attached to endoplasmic reticulum. Okay, so if the endoplasmic reticulum has these dots on them, the bumps, those are ribosomes. Okay, if it has those, it's rough. So the R in rough means it has ribosomes. If it does not, the endoplasmic reticulum does not have ribosomes on it, it's smooth. Okay? And they're just outside the nucleus. Okay, that's where those are. All right. Now, at the rough VR, or if it just finds a ribosome, that RNA, in the process of translation, is used to make protein. So DNA to RNA to protein is like one of the big things you want to get out of the chapter 2 and in chapter 3. Okay. So one thing I also want to clear up: DNA does not become RNA. Okay. RNA does not become protein. It's used to make. Okay. Used to make. All right. So also, the D in DNA, actually the N in NEA is for nucleic acid. RNA, it's also nucleic acid. Okay, so these are the same language, right? They're both nucleic acids. So when you make RNA, you're essentially making a transcript of this. So this is kind of like you're here at Panola, you're taking the classes. When you want to go on to a university or you want to go into nursing or whatever it is, you're going to have to show your transcript. This is your transcript, okay? So you take this and it's leaving the cell, or sorry, it's leaving the nucleus with it. Now, RNA to protein, that's a different kind of biomolecule, okay? Different class of biomolecule. So you're going to a different language. So you're translating. So I want to make sure you understand the difference between transcription and translation. Transcription is still in the nucleic acid category. Translation, you're going from nucleic acid to protein, okay? Another language. But that relates to the parts because, again, rough ER, and that's how you know the difference between the rough and the smooth ER. Okay? You might also see some ribosomes just floating around in your cell, and they can be used to make protein there too. All right, so we've kind of started the inside. I'm going to go to the out and then come back to the middle. This is your cell membrane, also known as the plasma membrane. I'll accept either. Okay? But I just want, if you see one or the other, know they're the same thing. What's a membrane? Anybody know what a membrane is? Okay? If you don't know what a membrane is, that's okay. All right? So a membrane is basically a barrier. Okay? It's a barrier between the inside and the outside of the cell. That's all it is. Okay? Keeps all the stuff inside in, keeps the other stuff out. So on the inside, well, actually, we'll stay with that. On the outside of this cell, you also have these little extensions called microvilli on the plasma membrane. Okay, these little structures, their role is to increase surface area. Okay? Because now that surface, instead of being flat, goes all the way around. Okay? Why is that important? These are for cells that need to absorb things. Helps you with absorption. Like in your small intestine, your food's been broken down. You gotta have a way to be able to take in those nutrients. They increase surface, the microvilli increase your surface area, and they help you absorb things. And that's what they do. All right. So, all right, let's go back in the cell. All right, so in terms of telling our story, you have your DNA, makes your RNA, that RNA leaves the nucleus, goes to the ribosome, makes protein. Okay, the protein might be a protein that needs to be used outside the cell. So in that case, those ribosomes at the rough ER make that protein. That protein goes to Golgi Apparatus. These are both, if you see that on those models, this is just like the outside, this is the inside of it, if you were to cut it open. Okay? So, kind of looks like inside of your slice open some flapjacks or some pancakes or something. But this is your Golgi apparatus. And that's for packaging and processing proteins. Okay? That's what they do. So, Golgi apparatus is kind of like Amazon. Amazon doesn't make the stuff. They basically, the companies send their stuff to Amazon, you buy it, they send it to you, right? Put it in the box, send it to you. So, here are the boxes. These are vesicles, these little circles, or spheres. Proteins end up in there, go to the end of the cell, or the edge of the cell, and they actually merge with the cell and release the contents, release the proteins out. And that's it's one of the ways that cells talk to each other. Okay, they send information out. All right. So that's your kind of story of DNA, RNA, and your protein. But wait, there's more, right? So, and I think we covered this, but the nucleolus makes your ribosomes or ribosomal RNA. Okay. Now, the nuclear envelope is just the border of the nucleus. Okay. Lysosomes, they break stuff down. Okay, they're like your cleaners. You know, like they're the cleanup crew. So if things are damaged, things are bad for you, lysosomes envelop them, and then they destroy them. Okay. Now, what else do we have? Mitochondria. So these look kind of like orange slices, but these, what they do is they make energy in the form of ATP. And we'll learn more about ATP in lecture, but it's, you know, we learned about chemical bonds and energy being in chemical bonds. ATP has some of those high energy chemical bonds in it. Okay, but that's what the role of the mitochondria is, to take nutrients like sugar and break them down to make ATP, make energy. Okay, so that's kind of like the story of the cell. Okay, at least day-to-day functions. All right, so all right, so if you want to, if you couldn't read my handwriting or you couldn't see it from back there, if you want to come up, I'm gonna probably erase this in just a second. So if you want to take a picture real quick, feel free. So I'll give you a minute to do that. So quick review. Where is the energy made in the cell? The mitochondria, all right? What part of the cell holds your genetic material, your DNA? The nucleus, okay? Where are proteins made in your cell? There's two places where proteins are made. Before you get to the Golgi apparatus, the rough endoplasmic reticulum and free in the cytoplasm. The other thing I didn't say was just cytoplasm. If it's just the space in the cell, that's cytoplasm, okay? So like all of this space in here, that's cytoplasm around all the organelles, okay? The fluid, we call that cytosol, but when it gets to be gel-like, we call it cytoplasm, okay? Now, I didn't mention the centrioles. So centrioles kind of look like little mini churros, right? They kind of have the little ridges on them, around them. I haven't really found a better example, but so I go with that. Their role is to help you with cell division, which is what we're going to talk about in a few minutes. Okay, so when a cell divides, the centrioles get on the opposite sides of the cell, and they start pulling the DNA chromosomes apart to opposite sides. So they're involved. Centrioles, and sorry, this is not on your thing, but in your notes somewhere, right? Centrioles and cell division. Okay, so just make sure you get that added. Okay, centrioles are for cell division. All right. Also, you also have cleaners, peroxisomes. Personally, I don't like trying to make you tell the difference between a lysosome and a peroxisome on a practical. But for all intents and purposes, these yellow circles are lysosomes. White circles are peroxisomes. They have very similar functions in terms of cleaning up. But just so you know, yellow circles are balls are lysosomes, the white ones are peroxisomes. If you see them on that model. Okay? All right. So what part of the cell packages and processes proteins? So it didn't make them, but it packages and processes them. Golgi apparatus. Okay? Golgi apparatus. Where's the ribosomal RNA made? The nucleolus inside the nucleus. Okay? Okay, just mention this. What part of the organelles are responsible or involved in cell division? Centrioles. Okay, good. All right, let's talk about some of these extensions. We just talked about microvilli. Remember we told you they were involved in absorption and increasing surface area? These are cilia. They're a little longer. Okay, so they're more finger-like. And they help brush things away. Okay, because they just kind of beat and brush things away. So you might have cilia, like in your nasal passages, cells there, as you get a lot of mucus and, you know, dust and things like that, that you want to just kind of push through. Okay, so they basically move things away. Okay, whereas flagella actually move that cell. All right, because the sperm cell is a cell. All right. And so here's our model. Okay, you all see this on practical. These are different stages of the life cycle of the sperm cell. But they all basically have flagella. Okay? Flagellum is singular, flagella is plural. Okay? But if you see this, probably flagella. Okay? So, see this, you right? See this, you right? Okay. All right. So, that's cellular extensions. Let's move on to cell division. All right. Cell, we just talked about cell's parts, but the cell actually has a life cycle just like people do. Okay? So, and believe it or not, the life cycle of a cell is really about 24 hours. Doesn't mean that all those cells are dead. It just means that's how long it takes before that cell is able to reproduce. Okay? So most of the time during the cell cycle, that cell is in what we call interphase. Okay? Interphase. And I can tell you right now, you're probably going to see a picture that looks just like this. And you're going to have to identify pieces of it. We'll just basically have all the letters and stuff, you know, blurred out. So just get used to seeing it, okay? So interphase has some main parts. You have G1 or gap 1. That's where the cellular contents are copied. Like all your organelles, your nuclei, your mitochondria, well, not so much your nuclei, but your mitochondria, your centrioles, your Golgi apparatus, endoplasmic reticulum, lysosomes, they get duplicated because you got to have enough for 2 cells. Okay? So that's where that stuff happens. Then we get to S phase. The S in S phase is for synthesis. What is synthesis? Making stuff. All right? It means making. So to synthesize something is to make it. What are we making? We're making DNA, genetic material. So we got to copy it. Okay? So in your lecture textbook, when you read your e-text or you do your dynamic study modules, for this, you'll see a term called replication. Okay? That's where that's happening. Okay? So just give you some framework, some context. All right. Then we hit the second gut phase, G2. And this is basically your final check, where you're checking your chromosomes, making sure your chromosomes aren't messed up. Okay, what are chromosomes? Chromosomes are DNA packed around protein, because it's easier to move that DNA if you tighten it up and pack. It's like if you move, you got to move out of your apartment. You got all your stuff all over your room. Is it going to be easier to move all that stuff with it just sitting there, or is it easier to put it all on a bag? All right, it's easy to put it all in some kind of suitcase or something first. That's what happens when we make the chromosomes. All right. So, but they got to check them, make sure they're good, right? Because you don't want any cells that's got problems, right? So, also, you have a little bit of cell growth that happens there. All right, this is your final checkpoint. All right, so then we're done with interphase. Then we hit M phase or mitotic phase. Okay. And so mitotic phase is sort of a broad category, which is mitosis and cytokinesis. And actually, at G2, we said this is a checkpoint. If there are problems, you can just go back to normal cell operation and no growth. Okay, it's basically like abort mission, right? We're gonna say, yeah, this one is not one you want to replicate, right? You don't want to make a copy of the cell, or you don't want to make a new cell because it's going to have bad DNA in it. Okay, so it just goes back to normal cell operation. All right, but if it's good, you go into mitotic phase. All right, and so then you have mitosis and cytokinesis. All right. So to review, most of the time the cell is in what phase? Interphase. What's the first part of interphase? G1, gap 1. What happens there? Well, before we hit DNA synthesis, what do we have to do? We got to copy all our stuff. We got to copy, and what? We got to copy all of our cellular contents, right? What's after that? After we copied our cellular contents, then what? We get to the S phase. What does the S phase mean? Replication, or what's another term for that? DNA synthesis. Okay. All right. So we got to make more DNA because you got to have two copies. All right. You have a copy for both new cells. After you made that DNA, then what do we do? Make sure your chromosomes are okay. All right. What do we call that phase? G2. Okay. If things are not good, what happens then? Where do we go? Call that G0. All right. That's the normal cell operation, no division, no growth. All right. Now, what if everything is good? Where do we go? We go to M phase, mitotic phase. All right. First part of mitotic phase starts with an M, mitosis. Second part is cytokinesis, which we'll learn about in a minute. All right, so now we're at the phases of mitosis. All right, first phase of mitosis is prophase. So you're gonna thinking about mitosis, use the acronym PMAT. So we're at prophase. Sometimes you'll see IPMAT, and we'll see Anaphase, of course, before that. Okay. All right. So during prophase, your DNA is kind of spread out. You still have It's starting to condense, starting to get packed up, but you still can see the nuclear membrane. Okay, so if you see a nuclear membrane, you're still in prophase. And then here are those centrioles we just talked about. Remember they said they were involved in cell division? Okay, they're starting to move to the sides of the cell. All right, by the time you get to late prophase, the centrioles have moved to the sides of the cell. And you get distinct chromosomes, no more nuclear membrane. Nuclear membrane's gone. So you can see that on the slide here. Here you can still kind of see nuclear membrane, but the DNA is still kind of spread out. All right. Here, DNA is starting to get more compact and you can't see a nuclear membrane. All right, but you can also see these centrioles on opposite sides. Then we hit metaphase. So when you think metaphase, think of the M in metaphase as middle. Okay, so the chromosomes line up in the middle. We call it the metaphase plate. So they were all kind of randomly organized. Now they're really organized in the middle. So you can see that on this slide as well. So if you see a slide, if you see a cell, and you can see that line, you know it's in metaphase. And yes, you will have to look at some slides on the practical. We have some for you to look at over there. All right. Anaphase. So we hit metaphase, now we're at anaphase. All right, so in anaphase, the sister chromatids of the chromosomes start coming apart. So the chromosomes split. And so you have two copies. Those two copies are, we call those chromatids. They split apart. So these are chromatids. So they split and start moving toward opposite sides. All right, you have these spindle fibers. They are attached to the centrioles. And they start pulling those sister chromatids to opposite sides. Then we get to telophase. And telophase, you can start to see a little line forming of two new cells. Okay, you also can start seeing nuclear membranes reforming. You can start seeing DNA start decondensing, start spreading back out. Okay, and that's what you see in telophase. So then we hit cytokinesis. Cytokinesis is where you start getting that distinct line. You get what we call a cleavage furrow in between these two cells. It's more noticeable than it is in telophase. But also, again, you really can see this nuclear membrane. And then DNA is all spread out again. Okay? Whereas here, you still see kind of a distinct area where the DNA is. Here, it's all spread out in little pieces. Okay? All right. Interphase, prophase, metaphase, anaphase, telophase. IP map. All right. Again, interphase. Okay, one cell with the DNA spread out. See nuclear membrane. Prophase, DNA starting to get more packed together. Okay. Metaphase, see that line? And again, this is however you see it on the microscope, because here the line's going down. You might see it at an angle. You might see it left or right. All right. It depends on the vantage point that you have for that cell. Anaphase, sister chromatids spread apart. Telophase, you're starting to get a cleavage furrow. Okay, you're starting to start developing that line. Okay. So if the chromosomes are in the middle of the cell, what do we call that phase? Metaphase. If the chromosomes are starting to move toward opposite sides of the cell, we call that? Okay. If you can still see a nuclear membrane, what do we call that phase? Well, you can see it a little bit in cytokinesis, but where do you really see it? In the very beginning. Which phase? Prophase. Okay. All right. Where do you start seeing a cleavage furrow in between? Telophase. Very good. Okay. So let's transition. So if y'all will make a line over here, and then we'll look at these slides.